being recorded.
Hello, everybody. Welcome to QUAHSI's Cyberseminar series. Happy Friday. Thank you for joining us today. My name is Kayla Berry. I am QUAHSI's Communications and Outreach Specialist. And I would just like to welcome our presenter today, Dr. Jan Willem Foppen. I also want to say thank you to Adam Ward, who is our Cyberseminar series host. He's with me every week, and he does a great job of introducing our speakers. As always, I like to begin with asking our participants to go ahead and type into the chat box where you're from, your affiliation. We like to get an idea of who is joining us and from where. So thank you as we get that conversation started. Right now I'm going to go ahead and pass the line over to Adam and let him introduce himself and also our presenter today.
Hi, thanks, Kayla. And again, I always like to recognize all the effort that you put in getting all of this technology to work for us and helping wrangle all of our speakers and their presentations each week. This week, I have the pleasure of introducing Jan Willem Foppen. He received his Master's of Science in Hydrogeology from the VU University in Amsterdam in 1990. He had a career as a consultant, living and working in Yemen for a number of years. And in 2004, he became a researcher at the UNESCO-IHE Institute for Water Education that's located in Delft. His research is primarily focused on the transport of colloidal particles in water, either surface waters or groundwaters. Much of his PhD work focused on the transport of E. coli through saturated porous media. Currently, he's looking at removal of E. coli, rotavirus, and adenovirus from contaminated waters flushed through chemically activated carbonized sand columns. And the reason we've invited him here today is about 3 years ago, he began the use of synthetic DNA tracers in surface water experiments as a way to be able to track colloidal particles through the environment. Additionally, Jan is the team leader of a research project focused on sanitation in slums of African megacities, looking at optimization of sanitation in urban slums in sub-Saharan Africa to reduce downstream eutrophication. He has been involved in teaching a number of courses and short courses, supervised nearly 30 Masters of Science students and 4 PhD dissertations, and that's just scratching the surface of the CV that I was able to take a look at before the talk today. So, Jan Willem, thank you very much for joining us. I'll turn things over to you, and we'll look forward to learning more about DNA tracers. Thank you.
Okay, thank you very much, Adam, for inviting me tonight. Here with me, it's tonight. It's about 9 o'clock in the Netherlands. So I'll be sitting in my attic and presenting this talk. Let me see. It's about the application of synthetic DNA in stream tracer injection experiments. That's the focus of the talk today. We do this together with Tom Bogaert from the TU in Delft and a number of students, and the most important ones for this talk are Judith Siopa, she's from South Africa, and Noel Bakobi, he's a student from Ghana. Table of contents, quite simple, straightforward background. I'll just try to give you a little bit of a background of what has been done before on DNA and using DNA as a tracer in the environment. What is the objective of today's talk? What have we done actually? Something on synthetic DNA. What is it made of? What are the dimensions of DNA and how to determine DNA concentrations in samples. What did we do? I'll just briefly talk about the methods that we used, the results and some conclusions, and then finally how can we improve what we have so far, and how can we hopefully apply in the near future this quite nice technique. It all started with a group in Norway, and Mr. Sabir, he was a Pakistani PhD student who was in Norway, and they injected DNA in the subsurface. Let me see, let me try to get an arrow here. How am I going to get this arrow? Ah, here is my arrow. So they had here an injection well in which they injected water. Here they had an injection point where they injected a slug of water spiked with synthetic DNA, and they pumped here for a number of days water out of this well, and between the pumping well and the point of injection they had a number of sampling points, and together with the DNA they also injected a salt tracer. After some time they found that here at these wells and these wells and these observation wells there were a lot of pluses, meaning that there they found DNA. This was a fractured aquifer somewhere in Norway, and that experiment seemed to work. They did a lot more of those experiments, and their conclusions after a number of field tests with synthetic DNA were that it's a very sensitive detection method. There is the availability of different sizes with variable valences of DNA. There's an infinite number of tracers. It's relatively cost effective. It's quite harmless to the environment. Well, actually it is harmless to the environment. It's a good alternative to organic or inorganic or radioactive tracers. But factors influencing the transport of DNA tracers like degradation and sorption, they need research. Around that time, Barbara Mahler she's now in the USGS, she experimented with DNA labeled monomerilonite in order to trace particle transport in rivers. And she added DNA labeled clay to jars which were filled with various water types, and after a number of days she PCR'd her samples, and she carried out gel electrophoresis, and then she identified her DNA particles back in her samples. The most important result of these experiments was that these DNA monomerilonite surface complexes are stable in waters for periods of up to 18 days, and they only slowly desorb to the aqueous phase. Their work, the work of Sabir and Mahler, had one major disadvantage, and that was that everything they did was qualitative. At that time, the technique to determine DNA concentrations was, you could only determine either the presence or the absence of DNA, but it was not possible to do anything quantitative. Thomas Ptak from Germany, from Tübingen, he was the first to do quantitative DNA work, and here you see an example of a laboratory column, or a breakthrough curve from a laboratory column, and this one here is bromide breakthrough curve, and the other one here is a DNA breakthrough curve. So they were actually able to quantify DNA concentrations, and that was simply because the technique to quantify DNA concentrations, quantitative PCR, became more and more available and became more and more affordable for laboratories throughout the world. A few years ago, we did in a couple of brooks in the Netherlands and in Belgium a number of stream tracer injection experiments, and this will be published in hydrological processes. And there we saw, when we injected DNA here, and we had 1 and 2 sampling locations in this little brook here, which is located here, we could quite easily trace back. Here, this is location 1 after 100 meter. This is location 2, 600 meters downstream of the point of injection. now we could relatively easily trace back DNA concentrations. And this here is the chloride breakthrough, and this here is our breakthrough curves of, 6 different DNA tracers. Last year, in ES&T, Sharma, he was the first to describe experiments in the environment whereby he used nanoparticles. And what they did was they had polylactic acid microspheres with a core of paramagnetic iron, that is iron which becomes magnetic when it is in the in the vicinity of a magnet, and they added to this microsphere a short strand of DNA. And their reasoning to do this was that this PLA, this polylactic acid, protects DNA from decay, and this paramagnetic iron is good to facilitate magnetic up concentration once you have derived your water sample. So here you have an example of an experiment that they did. This is what they call a plot experiment on an asphalt surface, and there they had the simultaneous and instantaneous application of 400 milligrams of tracer 1 and tracer 2. Here you have a garden hose, and they are near a parking lot. This is a flow dispenser, so water was flowing here, and it was ending here in this drain, and they applied here 2 meters from the drain tracer 1 and 1 meter from the drain tracer 2. They collected samples here, and here they measured breakthrough curves. Actually, when you read the publication, you'll find out that this here, the first tracer breakthrough, is tracer 1, but I think it should be the other way around. Now my arrowheads are not there anymore, because of course this is the one that breaks through first, and this is the one that comes after. Anyway, it seemed to work quite reasonably, although I think the quality of the breakthrough curves is not, it's not very, very beautiful. After our work in 2010 and 2011, we saw what we did, and everything was nice, but we thought, beautiful, but let's try to do it a little bit more quantitative and try to compare quantitative the behavior of just one synthetic DNA tracer and dissolved conservative salt, sodium chloride, in stream tracer injection experiments. And this is what we used. This is actually what we have used also in that publication of 2011. We have a tracer here which consists of 80 nucleotides. They're here. They're called bases or nucleotides. And of course DNA has a sugar phosphate backbone, and the length of an 80 nucleotide single-stranded DNA molecule is about 26 nanometers, and the width is about 1 nanometer. So now it's actually a kind of a small type of, colloid, basically. And something about the detection principle. So you inject the DNA, and how do you finally trace it back? Well, that's a little bit according to this principle. This is my synthetic single-stranded piece of DNA, and I add to that, to my sample, I add what is called a forward primer, which is a little piece of DNA. I add a probe, which is again a little piece of DNA, which has here fluorophore, and here what is called a black hole quencher. So as long as this quencher is in the vicinity of this fluorophore, the fluorophore doesn't give any signal. This here is the Ptak polymerase, which is the enzyme that duplicates my strand here, and after some time, my polymerase moves into this direction, it then starts to it starts to break down this probe, and when it does, the fluorophore is entering the environment and the environment of the sample, and once it is away from the quencher, it starts to give a signal. After some time, my duplication is completed, and then I also need to have a reverse primer to duplicate my strands in this direction. So what I add to one, what is called a qPCR well, is just 4 microliter of sample from my experiment or any other sample with an unknown DNA concentration. I add some chemicals, PCR buffer, some magnesium chloride. I add here forward and reverse primer and probe. I add the enzyme. I add the building blocks of my new DNA, ATP, GTP, CTP, TTP, and I add some water, and altogether I have about 20 microliter per sample. This I put in my machine, my qPCR machine, and I start to amplify it for 40 cycles. A cycle is defined by a denaturation step, which is something at a temperature of 95 degrees for 15 seconds, and then what is called an annealing extension step, which is at 60 degrees for 60 seconds. If I do that 40 times, I get a graph of, on the x-axis, the number of cycles, and on the y-axis, a relative fluorescence unit. The earlier this graph comes up, the higher the concentration of synthetic DNA in my sample. Then, when I know my sample fluorescence, I can compare it with a standard of known DNA tracer concentrations. Here, this is a standard, and standards typically look like this, with an R squared of about 0.999 and something. It's not that each and every piece of DNA is suitable for doing experiments. There are a number of sort of boundary conditions. First of all, the tracer and the primers, they should be unique. So that's something you can find out whether what you have designed is unique by going to the the NCBI. That's an institute of the National Institute of Health in the US of A, and there you can do what is called a blast search to find out whether your piece of DNA is known in the database. We chose to do 80 nucleotides. On the one hand, building DNA costs money, so it shouldn't be too big. Your piece of DNA, each and every nucleotide that you add, well, it has a cost, but it should also be unique. So you shouldn't have too little nucleotides. Your DNA trace shouldn't be too small. But 30 base pairs or 30 nucleotides is already quite okay and is already quite unique. Nevertheless, we chose our DNA tracers to have a length of 80 nucleotides. Then there are a number of parameters, like, okay, the length, that's 80 nucleotides. You should take care of what is called the GC content, the annealing temperature, and a few other things. And there shouldn't be any complementarities, there shouldn't be any similarity, etc. So there are a number of design considerations for good DNA tracers and primers. So what did we do this time? Well, we paid a lot of attention to that. I will not discuss that today. That's why I put it in these square brackets. We improved our protocol for taking samples, and we also used probes. In our previous work, we used a dye, which is called Cyber, but the disadvantage of that dye is that it amplifies each and every piece of DNA that is in your sample, and a probe usually does not. So a probe is more specific. Furthermore, the method that we use to determine concentrations is essentially an enzymatic method, and yeah, it's perhaps not very accurate. Also, we dilute a lot, and because finally you will be able, with your qPCR method, to detect 10 to 20 single pieces of DNA in a sample. And now, yeah, since you inject something like 10 to the power 14 or 10 to the power 15 pieces of DNA, yeah, you have a huge dilution, and you make errors in dilution. So we wanted to quantify those errors. We tried to make a mass balance of a number of DNA injection experiments in selected streams in the Netherlands, in Belgium, and in Luxembourg. And we did, so the characteristics of those experiments were that it was an instantaneous injection of a known DNA and a known sodium chloride mass. And then we, for each experiment, well, at least for this talk, for each experiment, these are the experiments that are included in this talk, that's what I wanted to say. We determined breakthrough curves of both DNA and sodium chloride at 2 locations downstream of the point of injection. Separate from these experiments, we did field batch experiments to determine decay, because you could lose DNA due to all kinds of reasons, and we had absolutely no idea what that could be, and whether decay was there or not. And we also carried out batch experiments with both river water and sediment to determine sorption of DNA to sediment. First of all, something on the dilution error analysis. Now, when you prepare a standard curve for qPCR, it is essentially the preparation of 13 tenfold serial dilutions. So this here, this final sample with a very low concentration of 10 particles per well, here after 30 cycles, is the 13th tenfold dilution. So you have a whole series of dilutions up to the original vial of DNA. Yeah, those serial dilutions, all of them, they are prepared with these relatively simple pipettes. And in total, you need about 36 pipetting actions to arrive at your dilution series. That's quite something. Those dilution errors, basically, and here we follow Hedges, who did a lot of work on dilution errors in bacteria counting. You basically have a pipetting error, and the measured accuracy or inaccuracy and precision of all our pipetting equipment was around 2% on a routine basis, which is well within the limits of what the manual says it should be. So the pipettes, they work well, but they just have an error. And in a vial, DNA particles are randomly distributed. So counts of DNA follow the Poisson rule, which means that the variance is equal to the mean, and the standard deviation is equal to the square root of the mean, but also that at each dilution you introduce a Poisson sampling error, and you need to quantify that. I will spare you all the difficult variance calculations. The most important thing here is this graph, and on the x-axis you have the nth tenfold serial dilution. So here we have our most diluted sample, which has been diluted 13 tenfold times. And on the y-axis is the ratio of the confidence interval at a 95% confidence level and the undiluted concentration. So what you see here is that due to dilution errors with your pipetting equipment and due to the Poisson sampling error, after dilution 5, you have an error, or let's say a confidence interval of about 15% of your original undiluted concentration. So the more you dilute, the larger the confidence interval becomes. And actually, when you are diluting this much, the confidence interval becomes about 40% of the undiluted concentration. So this is, we were actually a little bit surprised by the huge errors that you finally make with these also huge dilutions. The results of the field batch experiments. Here you have a graph of 3 batch experiments, and they were carried out in the field. And let me see, let me get my arrow here. So here and here and here are what was originally injected in the batch, which the batch was a bottle of a certain plastic. It was a polypropylene copolymer plastic, which is special and it doesn't absorb DNA. It's also known as a Nalgene bottle. I put those here to make a little bit of a distinction, but they should actually be here at time 0, so don't think they're here. No, they should be here at time 0. And so this we injected in, this was the Heuveler-Bach, one of the groups that we used, while we find here this concentration in our batch experiment. In the next 2 groups we have, this is what we inject, and these are the concentrations in our batches. From this we observe that first of all, this DNA mass in each of the containers was, it goes up and down just a little bit, but essentially it's more or less constant. So that was quite nice to see. Second of all, what we saw was that there is a huge difference between what we inject and what we find in our batch. And that difference, it started immediately. So within a few seconds, you'll see a huge difference between what you have injected and what you find back in your batch. And those Nalgene bottles, they were of course filled with river water, and we did those experiments immediately when we did our injection experiments in the brook. We did those experiments. Furthermore, the differences actually, this may be a little bit difficult to see, the differences between what is injected and what you find back, they are not constant. So this difference here is a bit bigger than this difference here, and then this difference here. So it's always a little bit different. Now here we have results with also batch experiments, but then with sediment. And you should know that you have a bottle which you fill with sediment, and that sediment, because it's quite coarse, these brooks, they have on their their river bottoms, the sediments are quite coarse, not too coarse, but let's say in the order of a few millimeters grain size of sand. So when you put that sand in water, it settles more or less immediately. And we shook that gently on a horizontal shaker to have a continuous sort of constant quality, constant mixing of DNA in our batches. And you see here that after 10 hours of doing that, we lose about 1 log here. So 1 log is more or less gone. one log of DNA. And here in this one, this brook, there seems to be a little bit of a decay, or well, disappearing of DNA particles, but not that much. And here there seems to be the DNA particles, they seem to disappear slightly. You should know that for the injection experiment that I will show in the next slides, we are always within, let's say, a few hours. So the experiments do not take long. Sometimes they take an hour, sometimes they take 2 hours, but not much more than that. So these conditions that we have here, we never encountered because our experiments were much shorter. Okay, so here is the summary of what I've just told you. So you can read it back later. And here we have the results of one stream tracer injection experiment, and this is in Sabir stream. It's a small stream which is located in Luxembourg, and at the time we measured it, it had a few tens of liters per second of discharge, not that much. Where's my arrow here? So what you see is that when we compare, this is sodium chloride and this is DNA. So here we have sodium chloride and here we have DNA, and watch those error bars because these are the confidence intervals that we have defined earlier from our error analysis. What you see here is that when you compare chloride and DNA, that the time of first arrival here, but also here, and also in the other experiments, which is actually the offset here from the y-axis, is more or less identical. Also, the shape of the curves going up and going down, in not every case, but they seem to be quite okay, at least for the rising part of the breakthrough curve. In a number of cases, the falling limbs of the breakthrough curves for salt were steeper than for, flatter than for DNA. The falling limbs of the breakthrough curve of DNA were steeper than of salt. So this is for the Maisbeek. This is another one for the Heulenbach, also in Luxembourg, this brook. And here you see that DNA is this one and salt is this one. And here they are quite okay, but here there is a deviation between DNA and salt. This is another example, and this here is salt, and this here is DNA, and there you see definitely that the DNA concentrations are much steeper. The breakthrough curve of the falling limb of the breakthrough curve is much steeper than of salt, and that's also the case here. And also you see here some funny outlier. So we think actually that's what we sometimes see. We see false positives. We call these false positives. And we think that these are compounds which are present in the river. They have taken, I don't know what is happening, but the Ptak polymerase, the enzyme that finally determines, are the legend label backwards? The legend labels, are they backwards? Oh, you're right. This is DNA. Yes, and this is salt. Correct. Apologies. This is DNA. Sorry, and this is salt. This is here DNA, and this is salt. Okay. My apologies, K. Brubaker. So we sometimes see here a false positive. We think that these are due to compounds which are present in the river, and they have sort of increased the Ptak polymerase activity. That is the enzyme responsible for for finally determining the DNA concentrations in our qPCR well. Okay. Yes, I have discussed this already. And let's see, here we have an overview of the mass balances of DNA and sodium chloride. So here we have our 3 experiments that I have just showed you. These are the discharges, and this is the mass of DNA that we injected. 10 to the power 15 to 10 to the power 16 particles of DNA. This is what we recover, and let me just show that to you like this. For the first experiment, we recover about 32% in location 1 and in location 2. For the next one, we recover some 6% in both location 1 and in location 2. And in the third one, we recover some 45 to 50% in location 1 and in location 2. So what we think we see here, and here, sorry, here you have the recovered sodium chloride masses. So they are quite high, although this one is a little bit lower, 85%, but these ones are close to what we have injected. Of course, there's always a little error in between injection and what is recovered. Coming back to the DNA concentrations, what we see here for each of these 3 experiments is that the mass there seems to be an instantaneous loss of mass, also in this case. And again, that loss was dependent on the brook in which that mass was injected. And what you also see is that per brook, the magnitude of the loss was more or less similar in both the injection and batch experiment. And furthermore, what you also see in this mass balance is that for 2 consecutive measurement points downstream of the point of injection, the DNA mass recoveries, they seemed to be more or less identical. So essentially what you see here, you also see on small scale in the batch experiments that we carried out. Yes, I more or less discussed this. Okay, I think I'm more or less almost there. What I wanted to show you was that we also tried to do mass balance modeling with OTIS. It's a little bit difficult to do this with OTIS because OTIS is not very good in these logarithmic concentrations, but nevertheless we did that, and the fitting that we did is fitting by eye, so it's not some sort of optimization scheme that we use. No, it's fitting by eye. And here we have for our 3 experiments, we have a set of parameters, and the parameters, dispersion, cross-sectional area of the brook, this is the exchange coefficient with the storage zone, this is the size of the storage zone. These are parameters that you typically can obtain with OTIS. They were for both sodium chloride and DNA breakthrough curves, they were the same. And then, in order to understand these steeper falling limbs of the DNA breakthrough curves, we added here a kind of absorption coefficient. This is what we have called, because that's one of the options in OTIS, the storage zone absorption coefficient. So in MySpeak, we needed that to have a better fit of model and measured data. In Heuvelbach, we did not need that, and in the B-lobe, we also needed such absorption coefficient. We also had to include initial losses, otherwise in the models we would have had too much DNA transported. So, conclusions. Compared to what we did earlier in 2011, we saw again that synthetic DNA performed as well as salt with regard to the shape and the timing of the breakthrough curves. We found that the DNA tracers, they covered a large concentration range of 4 to 5 log units. What was new was that the use of a probe, a Ptak probe, with a length of 25 nucleotides, increased the accuracy to detect synthetic DNA during qPCR a lot. So if you want to use this method, I can really recommend you to immediately start using a probe and don't do anything with Cyberseminar. The stepwise identification and application of a revised and improved protocol for sample taking and storage and tracer analysis further increased the accuracy of our entire method, and as a result we were able to quantify the error in the entire workflow. We found that both in batch experiments and after injection into the brook, DNA behaved in the same way. So first there was an instantaneous DNA tracer loss, and then the mass sort of became constant, at least for the duration of the experiment. Again, we found that for the duration of our experiment, the decay of DNA tracer, but also sorption, let's say main channel sorption, was insignificant. We were able to quantify longitudinal dispersion, transient storage zone dimensions, storage zone exchange rate, based on the combined use of sodium chloride and DNA tracer, DNA breakthrough curves. We're not yet there. The method is quite nice, but not perfect, definitely not perfect. So what we want to do in the near future is to find out causes and also to quantify the losses, the initial DNA losses that we found in our experiments. I'm not sure at the moment how we are going to do that, but this is definitely something which we like to have a better look at. We also would like to reduce the occurrence of these false positives, because every now and then you have them, and it's quite annoying. So how can we get rid of that? Perhaps some sort of filtering. We have played a lot with all kinds of filters, with cellulose acetate filters, with glass fiber filters, but all these filters, they change DNA concentrations, and cellulose acetate filters, they even they cause the Ptak polymerase to work much faster, and concentrations become much higher than what they really are. So do not use cellulose acetate filters in qPCR. It's not a good idea. And what we have not done at all really is to explore the long-term behavior of synthetic DNA tracer, because if you want to do longer-term experiments than what we have done, I can imagine that there is a DNA loss, and that needs to be quantified, of course. And perhaps the route that Sharma took in his work in ESNT in 2012 is a very nice way forward, and also a very elegant way forward. Although his breakthrough curves are maybe not very beautiful, that's just something, perhaps if you work on it, then things will improve dramatically. The advantage of nanoparticles is that you protect your DNA from decay. At least that's what you expect. Some future applications. I think with the way that we use DNA at the moment is we are able to determine dispersion, and we did some calculations with in spreadsheets with okay, how high can the discharge be before you dilute away completely your DNA concentration. That is something in the order of 100 cubic meter per second, and even more before you completely lose your DNA signal. So it's actually quite difficult to completely lose your DNA signal. The enormous dilution and also the enormous low concentrations that you can measure, up to 20 particles of DNA, is really fantastic. I think with the current state of the method, it is possible to determine source areas and to analyze flow paths in, for instance, complex flow systems, also to do multi-tracer applications, flow systems like karstic areas, or like also Sabir already did 20 years ago in fractured aquifers. Perhaps it's possible to do something on sediment transport processes, so then it becomes a kind of a particle tracking velocimetry. And hopefully in the future it is possible to have really quantitative multi-tracer applications in, not everywhere, but in selected surface water and groundwater applications. That is at least our vision. Thank you very much.
Well, thank you very much for a great talk. This is Adam. And one of the awkward bits of this is that although people are applauding really all around the world, I think, from who's called in here, you don't get to hear them through the Adobe Connect software.
That's okay. That's okay.
But again, thank you, and thanks for leaving some time for questions and answers. So for folks on the line, if you do have questions, I would ask that you type in the chat box at least, hey, I have a question, and I'll do my best to moderate these things, and you all can sort of unmute yourselves, and we'll just try to address these in the order at which they're asked, if that's okay. And so, Diogo at Notre Dame, I see you've got both a hand raised and you were quick on the question. So go ahead, please.
Hi, thanks. Nice talk. Really quickly, in the batch experiments, what is it that causes the instant degradation?
Thank you for that question. You see, we do not know, actually. One idea is that low concentrations of trivalent ions in river water, I mean, these are, environmental water, so you do not know what is in there. You cannot really analyze those waters for 100% up to the nanomole, but we think that certain ions in those waters cause losses, because what we see is that in various streams, in different streams, we have different losses. And when you do these experiments in demi-water, you have no losses at all. So you can trace back everything that you inject. There's no problem whatsoever. But when you do this in environmental waters, you have a huge loss. So we think that it is due to the chemistry of these streams.
Thank you.
Okay. Oh, Diogo, did you have a follow-up? Sorry.
No, just thank you.
Let's see. The next one I see is a question from Roy Haggerty. Roy, go ahead.
Hi, thanks for the talk. Let's see. So I noticed that in the breakthrough curves, you had relatively few breakthrough curves relative to obviously what you'd have in a if you had an in-stream analyzer of some kind. You have to take samples and so on and so forth. And that indicates a fairly high cost. So I'm wondering what kind of time it takes to analyze a sample, or maybe you could give the answer in terms of tens of samples or hundreds, whatever you want to do. And then also in terms of limitations of, in fact, costs in terms of euros, dollars.
Mm-hmm. Thank you for this question, Mr. Haggerty. We can do with our qPCR machines that we have in our laboratory around 400 samples per day. And the cost of one sample is in the order of if you take the sorry, I see a question here, another chat there. If you take the right compounds and the right consumables, there are huge cost differences. But if you do it in a smart way, then it is possible to do it for only 10 to 20 cents per sample.
Okay, then just quick follow-up. So I see, in your breakthrough curves, they typically have 15, I don't know, it looks like 15 or so samples. So why, sorry, I don't mean to be critical in this way, but...
it's okay, no problem.
Why so few samples?
Why so few samples? I think we have 2 or 3 times more samples still in our freezers in the laboratory. There could be more, but we thought that this number of samples that we have analyzed, that they were already quite sufficient to show the principle.
Okay, so basically you're just saying, and I agree with that. The limitation here is not in cost or effort then?
Absolutely not.
Yeah, okay, good, thank you.
All right.
Let's see, so we've got a question in the chat box from Ashley. She wants to know some more about how you collected your stream samples, and do you have to concentrate them prior to qPCR?
We collect them immediately from the brook with a Gilson pipette man. So we have our gloves on. We do not touch the brook at all. We have disposable, how do you call these things? Disposable filter tips with low adhesion characteristics. We take 1 milliliter of sample, and we then fill an Eppendorf vial, a 2 milliliter Eppendorf vial with 1 milliliter of sample, and we then immediately store it on dry ice.
Okay, let's see. Temple University Geology wonders if the tails of the breakthrough curves appear different, why did the sodium chloride and DNA tracers calibrate to the same parameters in OTIS?
Because the dispersion and so let's say the dispersion and the cross-sectional area of the brook are more or less defined by the rising limb of the breakthrough curve, and this is, we have another one also. So here in this case, this is another one. You see that the falling limb, to a certain extent, but not everywhere, follow each other, and then you just need one little additional parameter to explain these steeper curves. And mind you, we are here already at very low concentrations. So, and it is indeed curve fitting by eye. So, in that respect, we surely can do better, because there are more and better pieces of software available to do something in this logarithmic space of our concentrations.
Okay. May I jump in? I'm not sure if there's another question. It's Roy.
Yeah, go ahead, Roy.
Jan, if you're interested, and a couple of other people here online, or 4 maybe of us have used it, you're welcome to use some software that I wrote, Stamped-L. It's designed basically for these kind of log tails. If you're interested, I'll be happy to send it to you.
I would be happy to receive that.
And it also does reactive tracers and things like that. So probably
I would be really happy to receive that. What is the name of that software, please?
Oh, it's got a goofy name. It's Stamped-L, STAMPT-L. I'll send you the code right after this.
Is that something I can download from your website?
It's not on the website. I try not to be a software manager, but I release it to anybody who wants it, and be happy to send it to you with user manual and everything.
I would really like to have that.
Okay.
Thank you very much.
Yep, you bet.
Do we have any other questions from folks who are on the line today?
Could you send me the code too, please?
Yeah, sure, Diogo.
Sorry.
I'll make just one announcement. This is Adam. If you've been watching the chat box, you saw this scroll by. As Kayla mentioned before we started the talk, she does record all of these, and with a few days of processing, these are on the website. So if you visit QUAHSI's homepage next week, you should be able to find your way to a recording that we archive indefinitely of all of our cyber seminar talks, including this one.
Nice.
I'll give one last call here for any questions from folks who are on the line. Okay, well, hearing none,
sorry, Adam, just saying, Jan, I mean, I know your name and so forth, but your email address, is it easy to find online?
Let me see. Is my email address here? It should be.
Put into the chat box, Jan.
Here. Here's my email address.
There you go.
j.foppen@UNESCO-IHE. Got it.
All right. Thank you.
All right, Jan. Well, I also wanted to thank you again for taking the time out of your evening to talk with us today. It was a great presentation, and we look forward to working with you in the future.
I enjoyed giving this presentation. Thank you for the invitation.
You're very welcome. Well, thank you, everybody, for joining us, and we look forward to your participation next week. Thank you, Adam.
Yep, thanks, everybody, for attending, and we'll see you a week from today for our next seminar. Thank you.
All right. Goodbye.
Goodbye.